Beneficial prokaryotes. Not all prokaryotes are pathogenic. On the contrary, pathogens represent only a small percentage of the diversity of the microbial world. In fact, our life and all life on this planet would not be possible without prokaryotes.
Welcome back to Concepts of Biology. We'll continue to read section 13.1.
Prokaryotes and Food and Beverages. According to the United Nations Convention on Biological Diversity, biotechnology is any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. The concept of specific use involves some sort of commercial application. Genetic engineering, artificial selection, antibiotic production, and cell culture are current topics of study in biotechnology. However, humans have used prokaryotes to create products before the term biotechnology was even coined. And some of the goods and services are as simple as cheese, yogurt, sour cream, vinegar, cured sausage, sauerkraut, and fermented seafood that contains both bacteria and archaea. Figure 13.9. Some of the products derived from the use of prokaryotes in early biotechnology include cheese, salami, yogurt, and fish sauce. Cheese production began around 4,000 years ago when humans started to breed animals and process their milk. Evidence suggests that cultured milk products like yogurt have existed for at least 4,000 years. Using prokaryotes to clean up our planet. Bioremediation. Microbial bioremediation is the use of prokaryotes or microbial metabolism to remove pollutants. Bioremediation has been used to remove agricultural chemicals, pesticides, and fertilizers that leach from soil into groundwater. Certain toxic metals, such as selenium and arsenic compounds, can also be removed from water by bioremediation. The reduction of selenate to selenite and metallic selenium is a method used to remove selenium ions from water. Mercury is an example of a toxic metal that can be removed from an environment by bioremediation. Mercury is an active ingredient of some pesticides. It is used in industry and is also a byproduct of certain industries, such as battery production. Mercury is usually present in very low concentrations in natural environments, but it is highly toxic because it accumulates in living tissues. Several species of bacteria can carry out the biotransformation of toxic mercury into non-toxic forms. These bacteria,
such as Pseudomonas aeruginosa,
can convert mercury iron to elemental mercury, which is non-toxic to humans. Probably one of the most useful and interesting examples of the use of prokaryotes for bioremediation purposes is the cleanup of oil spills. The importance of prokaryotes to petroleum bioremediation has been demonstrated in several oil spills in recent years, such as the Exxon Valdez spill in Alaska in 1989, the Prestige oil spill in Spain in 2002, the spill into the Mediterranean from a Lebanon power plant in 2006, and more recently, the BP oil spill in the Gulf of
Mexico in 2010. To clean up these spills, bioremediation is promoted by adding inorganic nutrients that help bacteria already present in the environment to grow.
Hydrocarbon-degrading bacteria feed on the hydrocarbons in the oil droplet, breaking them into inorganic compounds. Some species, such as Alcanivorax bercumensis, produce surfactants that solubilize the oil, while other bacteria degrade the oil into carbon dioxide. In the case of oil spills in the ocean, ongoing natural bioremediation tends to occur inasmuch as there are oil-consuming bacteria in the ocean prior to the spill. Under ideal conditions, it has been reported that up to 80% of the non-volatile components in oil can be degraded within 1 year of the spill. Other oil fractions containing aromatic and highly branched hydrocarbon chains are more difficult to remove and remain in the environment for longer periods of time. Researchers have genetically engineered other bacteria to consume petroleum products. Indeed, the first patent application for a bioremediation application in the US was for a genetically modified oil-eating bacterium. Figure 13.10. Cleaning up oil after the Valdez spill in Alaska. The workers hosed oil from beaches and then used a floating boom to corral the oil, which was finally skimmed from the water surface. Some species of bacteria are able to solubilize and degrade the oil. One of the most catastrophic consequences of oil spills is the damage to fauna.
Prokaryotes in and on the body. Humans are no exception when it comes to forming symbiotic relationships with prokaryotes. We are accustomed to thinking of ourselves as single organisms, but in reality, we are walking ecosystems. There are 10 to 100 times as many bacterial and archaeal cells inhabiting our bodies as we have cells in our bodies. Some of these are in mutually beneficial relationships with us, in which both the human host and the bacterium benefit, while some of the relationships are classified as commensalism, a type of relationship in which the bacterium benefits and the human host is neither benefited nor harmed. Human gut flora lives in the large intestine and consists of hundreds of species of bacteria and archaea, with different individuals containing different species mixes. The term flora, which is usually associated with plants, is traditionally used in this context because bacteria were once classified as plants. The primary functions of these prokaryotes for humans appear to be metabolism of food particles that we cannot break down, assistance with the absorption of ions by the colon, synthesis of vitamin K, training of the infant immune system, maintenance of the adult immune system, maintenance of the epithelium of the large intestine, and formation of a protective barrier against pathogens. The surface of the skin is also coated with prokaryotes. The different surfaces of the skin, such as the underarms, the head, and the hands, provide different habitats for different communities of prokaryotes. Unlike with gut flora, the possible beneficial roles of skin flora have not been well studied. However, the few studies conducted so far have identified bacteria that produce antimicrobial compounds as probably responsible for preventing infections by pathogenic bacteria. Researchers are actively studying the relationships between various diseases and alterations to the composition of human microbial flora. Some of this work is being carried out by the Human Microbiome Project, funded in the United States by the National Institutes of Health.
Hi! If you found this video informative, share the link. We want these free textbook resources to be available to other students. Alright, let's get back to reading.
Now on to Section 13.2, Eukaryotic Origins.